Welcome back to the deep dive. Sometimes you look at these really dense scientific papers and just wish someone could, well, cut through it all and just tell you what's really important. That's exactly what we're aiming to do for you today. We're diving into some fascinating new research. Our focus today is a study called Muscle Biopsy Findings in Valosin-Containing Protein Multisystem Proteinopathy. That first word is Valosin, rhymes with Palosin, or VCP-MSP for short. It's brand new research just out.
That's right, Schiava and colleagues 2025. And it really digs into what muscle biopsies show us about this condition.
So our mission here is to pull out the key insights, the really important stuff, specifically from looking at the muscle tissue itself. What does it tell us?
Exactly. And what's really interesting is how the study tries to comprehensively look at the muscle pathology, the myopathology, and then connect that to how patients present clinically, their specific genetic variations, even how the disease might progress over time.
Yeah, and the scale of this thing really jumped out at me. This wasn't like a small local study.
No, not at all.
It was huge, a retrospective international effort. They pulled together biopsy images, data from 112 adults, all confirmed VCP variants, right? And from, get this, 52 centers in 24 different countries.
Wow. That kind of scope, that's what gives the findings real weight, doesn't it?
Absolutely. So just to give you a quick snapshot of who was in the study, mostly male, about 2/3. Mean age when the biopsy happened was around 53. And they'd had the disease on average for about 6.5 years.
Okay. Any particular genetic variants stand out?
Yeah, the most common one, technically called C.464GA or PARG155-His, that was found in almost 19% of them.
Right, ARG155-His. That's a pretty well-known one in VCP-MST.
And clinically, how did it usually show up?
Well, the study found isolated myopathy, so just muscle weakness was most common, about 38%. Then you had myopathy combined with Paget disease of the bone, that was around 18%. And myopathy with motor neuron problems, like ALS-type symptoms, that was about 13%. Shows the variability right there.
It really does. So before we dive into the biopsies, maybe just a quick refresher on VCP itself. What does this protein normally do?
Sure. So Valosin-containing protein, VCP, it's fundamental, really crucial for keeping cells healthy, maintaining homeostasis. Think of it as part of the cell's quality control and waste disposal system. It's involved in degrading old or damaged proteins. Also in autophagy, that's like the cell's recycling program controlling the cell cycle, even regulating programmed cell death or apoptosis.
So it does a lot of important jobs.
A lot. And VCP-MSP happens when you get these pathogenic variants, these sort of errors in the VCP gene. And that just messes up all these vital functions, leads to, well, cellular chaos, essentially.
Okay, got it. So now the really interesting part, the detective work. What did the researchers see when they looked at these muscle samples under the microscope?
Right. This is the core of the study. They found some pretty consistent things across the board. Most frequent. Atrophic fibers. That just means muscle fibers were shrinking. Seen in almost 90% of the biopsies.
Shrinking fibers. And what did that look like? Just smaller.
Yeah, they describe them as either looking kind of angulated, pointy almost, or just generally small and rounded. Yeah. And usually it affected both the main fiber types, type I and type 2. That was seen in nearly 3/4 of cases with atrophy.
Okay, shrinking fibers. What else?
Rimmed vacuoles. These are quite characteristic, though not unique to VCP, found in about 72% of cases.
Rimmed vacuoles. Can you describe those a bit more?
Imagine tiny little bubbles or spaces, often with a distinct border or rim, appearing inside the muscle fiber. It's a sign that things aren't right internally, that stuff is accumulating maybe.
Okay, like little pockets of trouble inside the cell.
You could put it that way, yeah. Then endomysial fibrosis, that was in 58%.
Fibrosis, that's like scarring, right?
Yes, an increase in fibrous connective tissue within the muscle. Makes it stiffer, less functional.
And the last big one.
Protein aggregates. Clumps of abnormal proteins, basically. Found in just over half the biopsies, 51.8%.
Protein clumps. Did they know what proteins were clumping up?
Yes, they looked into that. The main ones were a protein called P62. That was in about 60% of the aggregates they analyzed. And interestingly, VCP protein itself was often found in these clumps, in about 36% of them. Also things like myotilin, desmin, other muscle proteins.
Wow, so the VCP protein itself is part of the problem clump. That seems significant.
It really is. It points directly back to the VCP dysfunction we talked about.
Now, there was something else you mentioned, a unique finding they called degeneration niches.
What?
That sounds intriguing.
Yes, these were quite distinct. They saw them in about 31% of biopsies. These weren't just scattered shrinking fibers. There were specific localized areas.
Like little pockets of decay.
Kind of. Demarcated zones where the muscle structure was really altered. You'd see severely shrunken atrophic fibers, all embedded in this mix of fibrotic scar tissue and even fatty tissue.
And these could be sitting right next to normal muscle.
Exactly. Often found within areas of otherwise pretty normal-looking muscle tissue. And importantly, they saw these niches across all the different clinical presentations. The isolated myopathy, the ones with Paget's, the ones with motor neuron signs.
So it seems like a core feature of the muscle damage process itself, maybe.
That's what it suggests. A very specific, localized way that damage happens or spreads in the muscle.
So how does all this, the shrinking fibers, the vacuoles, the aggregates, these niches, how does it all tie back to VCP not working correctly?
Well, it connects quite directly. Normally, VTP forms this complex, like little machine, a hexamer. And its job includes grabbing onto proteins that are marked for destruction, often tagged with ubiquitin and helping escort them to the cell's disposal systems, like the proteasome.
Okay, the cleanup crew.
Right. But when VCP is faulty, like in VCP-MSP, this process breaks down. It can't efficiently clear out the junk proteins. So these ubiquitinated proteins accumulate.
Yeah.
Where do they often end up? Inside those rimmed vacuoles we saw.
Ah, so the vacuoles are like little trash bags filling up because the garbage truck isn't working.
That's a decent analogy, yeah. And also, faulty VCP can impair autophagy, that recycling process. So things that should be recycled, like the protein P62, which acts as a cargo receptor for autophagy, also build up. That explains the P62 aggregates.
It all starts to make sense at the cellular level. And now, did the researchers see a pattern in how severe these changes were? Could they sort of map out how the muscle damage progresses?
They did observe a real spectrum of severity, which allowed them to infer a possible natural history. Yeah. So in the mildest cases, maybe just a few scattered abnormal fibers, early signs.
Just starting out.
Right. Then in more advanced stages, you see larger areas, these foci of atrophy and damage. Those fibers get progressively replaced by that fibrotic tissue and fat we mentioned. And in the most severe cases, the overall tissue structure is really disrupted. But interestingly, even in some very late-stage samples, they could still find pockets of relatively preserved, normal-looking muscle.
So it's not necessarily a uniform wipeout of the muscle. It can be quite patchy.
Exactly. Patchy and progressive seems to be the pattern.
Okay, let's connect the dots then. We have these microscopic findings. How do they relate to the patient's specific genetic variant or their clinical symptoms? Were there clear correlations?
This is where it gets really interesting and maybe a bit unexpected. One of the biggest takeaways was that, for the most part, there were no significant differences in how often they saw these biopsy findings, the atrophy, vacuoles, etc., among the 4 most common VCP gene variants.
Really? So different genetic misspellings mostly led to the same picture under the microscope?
Largely, yes. With one notable exception we should point out.
Okay.
Those degeneration niches we talked about. They were absent in the biopsies from 12 patients who all had a specific variant called PRs93Cs.
So that variant seems different in terms of the niches. But otherwise, the common variants looked similar microscopically.
Pretty much. Which suggests that maybe the way the VCP protein malfunctions downstream leads to a similar kind of cellular damage pattern, regardless of the specific initial genetic hit, for many common variants at least.
That's quite counterintuitive. You might expect more variation. What about matching the biopsy to the clinical picture? Did they line up well?
That's the other surprising part. Often they didn't line up perfectly. They found that clear agreement or concordance between the clinical presentation, the biopsy findings, and things like nerve conduction studies only happened in about half the cases, just 49.4%.
Less than half. Wow.
Yeah, it really highlights what they called significant heterogeneity, how varied this disease is. For example, remember those patients who clinically only seem to have muscle problems, isolated myopathy?
Right, the biggest group.
Well, in 20% of those patients, the biopsy actually showed signs of both muscle damage and nerve involvement, a mixed pattern.
So the biopsy was revealing problems that weren't obvious from the symptoms alone.
Precisely. It suggests the underlying pathology can be broader than the clinical presentation at that snapshot in time.
Did they look at MRI scans too?
They did mention it briefly. For patients who had an MRI, about 47% showed fatty infiltration, like fat replacing muscle. And they speculated these fatty areas on the MRI might actually correspond to those degenerative areas, the niches or larger foci seen in the biopsies.
Okay, so bridging the microscopic to the macroscopic imaging makes sense. So for you, our listener, why dig into all these microscopic details? What's the practical takeaway here?
Well, the big one is diagnostic value. These consistent patterns, even if not unique, are really characteristic of VCP-MSP. So if you have a patient, maybe without a known family history, presenting with muscle weakness and maybe some other features, seeing this combination of rimmed vacuoles, P62 aggregates, maybe those niches, and relatively little inflammation, that can strongly point the clinician towards testing the VCP gene. It helps differentiate it from other muscle diseases.
So it provides important clues for diagnosis.
Definitely. But, and this is really important, but these findings are not pathognomonic.
Meaning they aren't only found in VCP-MSP.
Exactly. Rimmed vacuoles, for instance, are famously seen in other conditions like inclusion body myositis, IBM. That's a much more common muscle disease in older adults.
So how do pathologists tell them apart if they both have rimmed vacuoles?
There are other clues. One key difference highlighted in the study and known in the field is inflammation. In IBM, you typically see significant inflammation, including widespread expression of something called MHC class 1 on the muscle fibers. But in VCP-MSP, that MHC 1 expression is usually very limited, maybe just on a few regenerating or degenerating fibers. It's generally not an inflammatory myopathy in the same way IBM is. That subtlety is crucial for diagnosis.
Got it. So it's the whole picture, not just one finding.
Precisely. Context is everything.
And we should probably mention, like with any study, there were limitations. Right.
Of course. It was retrospective, meaning looking back at existing data, which always has challenges. Different centers might have used slightly varied staining techniques over the years, and for some specific features, like comparing the degeneration niches to other diseases, they didn't have ideal comparison groups within this data set.
Acknowledging those limitations is just part of good science.
Absolutely. It helped frame the findings properly.
Was there anything about future research directions, any intriguing ideas that came out of this?
Yes, actually. Relating to those degeneration niches again, the authors put forward a really interesting hypothesis. They speculated that maybe the way these damaged areas seem to expand could involve some kind of cell-to-cell communication between muscle fibers, perhaps similar to mechanisms seen in prion diseases, where misfolded proteins can sort of seed aggregation in neighboring cells.
Wow, like the damage could spread from cell to cell.
It's a hypothesis. Needs much more investigation, of course. But it's plausible because VCP is known to be involved in controlling protein aggregation, even suppressing seeding mechanisms in neurons in other contexts. So maybe when VCP fails, this suppression is lost in muscle too.
That's a fascinating angle for future work. So wrapping this up, this deep dive really underscores something important, doesn't it? That even though VCP-MSP can look different clinically and come from various genetic variants, when you look closely at the muscle tissue itself, there are these consistent, characteristic, microscopic features.
Exactly. And those features give us vital clues about what's going wrong biologically, the pathophysiology. And critically, they help doctors diagnose the condition more accurately. It shows the power of pathology.
It really does. It's connecting the gene defect to the cellular chaos to the patient's experience.
Which leads to maybe a final thought for you to consider. In this era of really complex diseases where patients can present so differently even with the same underlying cause, how vital is it to integrate everything? The genetics, the clinical picture, and these really detailed cellular findings from pathology. This study really shouts that sometimes the deepest answers aren't just in the broad strokes, but hiding in those precise microscopic details.
A powerful reminder. And hopefully exploring those details today helps you piece together the puzzle of this condition a little better, making you even more informed.